Hi everyone. In this video, we are going to discuss about the structure and the function of Megasporangium. We all know that gynoecium is the female reproductive part of the flower, and gynoecium is having three parts. That is, the uppermost flat part is known as stigma. Then we have a long and thin stalk, which is called style. And at the bottom of this gynoecium is a bulged or swollen part present, which is called ovary. Okay, and we have already studied this in the previous videos that this ovary encloses ovules inside them. Okay, so what is present here? This ovary encloses ovules inside them. Okay, so this megasporangium is commonly known as ovules. Okay, so megasporangium and ovules, they both are same. That means in this video we are going to learn about the structure and function of these ovules. Okay, the number of ovules present inside ovary actually varies from species to species. Okay, and these ovules inside ovary are actually attached to the ovary wall with the help of a tissue which is called placenta. So placenta is having two functions. First one, it actually helps in the attachment of ovules to the wall of ovary, and second one, the cells of placenta are nutritive in function. They actually provide nourishment to the developing embryo present inside the ovules of this ovary. Okay, and we have studied this in grade 11 that there are different types of ovules in the flowering plant, and these are like anatropous, orthotropous, hemitropous, campylotropous, etc. Okay, so the most common type of ovule present inside the flowering plant or the angiosperms is anatropous ovule. Okay, the type of ovule which is commonly present in most of the angiospermic plant is anatropous. So anatropous ovule is the one which is present completely upside down. Okay, it is present completely upside down. It takes 180-degree turn. Okay, and the meaning of this bite technique I will tell you a little later after introducing few parts of the ovule. Okay, now on the right side this picture is given. For example, this is the ovary wall. Okay, this is the ovary wall. Okay, and we have a tissue called placenta. So this ovule is actually attached to ovary wall with the help of a tissue called placenta. Okay, now from this placenta a stalk arises. Okay, this stalk is known as funicle. Okay, so this ovule is having two parts, funicle and the stalk part, which is called funicle, and the body part. Okay, now this body is actually arising from the funicle at this particular region. Okay, so this stalk part is known as funicle and the lower part is known as body of the ovule. Okay, and the junction between this funicle and the body of the ovule is known as hilum. Okay, and in the body of this ovule you can make out that in ovules different layers of cells arises. Okay, and these layer of cells actually form the outer covering of embryo sac. We all know that inside ovule female gamete is present inside the embryo sac. Okay, so this embryo sac is actually enclosed, present deep inside the ovule. Okay, so this outermost layer of cells present inside the ovule, they form a kind of protective covering which is known as integuments, and there are two integuments, outer integument and inner integument. Okay, inside integuments there is a mass of cell present which is called nucellus. Okay, and this nucellus inside it encloses the embryo sac. Now the part of the ovule, you can say the base of the ovule, from where actually this integuments appears to start. Okay, the part of the ovule from where this integument layer actually appears to start is known as chalazal pole. Okay, and the part of the ovule which is opposite to this chalazal pole, directly opposite to this chalazal pole, is known as micropyle. Okay, and this area where actually this integuments are absent, because integuments are present all around the embryo sac except at the tip of this embryo sac or tip of the ovule. Okay, so this point is actually known as micropylar pole. Okay, or just this opening is called micropyle. I will explain this to you once again by drawing the diagram. For example, this is the ovary wall, okay? And you have a tissue which is called placenta. So this is ovary wall and this is placenta tissue, okay? Now attaching to this placenta, starting from this placenta is the stalk of the ovule, which we have already discussed, okay? The stalk of the ovule is known as funicle, okay? And then arises the body of the ovule like this, okay? And we have already seen in the previous slide that this body of the ovule is having a layer of cells, okay? This layer of cell is present as a protective covering around the ovule, okay? In the ovule, the outermost layer of cells is present as a protective covering around the embryo sac, okay? And this part, funicle, is actually known as the stalk of the ovule, and rest part of the ovule is actually known as body of the ovule, okay? Body of the ovule. So the junction between the stalk and the body of the ovule is known as hilum, okay? So the junction between the stalk and the body of the ovule is called hilum, okay? And this end of the ovule, from where these integuments appear to arise, or from where these integuments actually appear to start, is known as chalazal pole, okay? And the end which is directly opposite to the chalazal pole is called micropyle, okay? And in the center of this ovule, there is a mass of tissue present, which is known as, there is a mass of tissue, or there is a mass of actually cell present, and this mass of cell is known as nucellus, okay? And these layers of cells, protective layers of cells, are outer integument, and then we have inner integument, okay? So initially these are the parts of the ovule. Micropyle is the end of the ovule where integuments are absent, okay? And these are the parts of the ovule in an newly formed ovule, or the developing ovule, okay? Now slowly in later stages what happens is, one of the cell of the nucellus, okay, remember this point, one of the cell of the nucellus actually develops, it enlarges and develops into the megaspore mother cell, MMC, okay? So here the meaning of MMC is megaspore. Megaspore means ovum, okay, the female gamete. So the cell which will actually produce female gamete is known as mother cell. So megaspore mother cell, okay? And this megaspore mother cell is actually diploid, okay? Obviously it will be diploid. It will undergo meiosis 1 and meiosis 2, and it will produce haploid gametes, okay? So this megaspore mother cell is diploid, okay? Now what happens is, in this ovule you can observe there are two integuments present, outer integument and inner integument, okay? So that's why in angiosperms all the ovules, they are known as bitegmic ovule, okay? Bitegmic. The meaning of term bi is two, and tegmic means tegments, okay? So that means the ovules which contain two integuments, that's why they are called bitegmic ovule. So in angiosperms we have ovule which is anatropous and bitegmic, okay? In all the gymnosperms, this is given in the previous slide, in all the gymnosperms we have unitegmic ovule, okay? Uni means one and tegmic means integument. That means the ovules of gymnosperms, they have only one integument present around them. Fine. And this nucellus tissue which is present inside the center is actually, it's highly nutritive in function. It provides nourishment to the developing megaspore or to this developing embryo sac, okay, to the developing megaspore or to the developing embryo sac, okay. And in some ovules, nucellus is present, only it's not present, it's not compactly packed, okay. The cells of the nucellus is not compactly packed, it is present only in half form. That means only half of this area is filled with the nucellus. So this type of nucellus or this type of ovule is known as Tenuinucellate ovule. Okay, that means ovule in which only some part of nucellus is present, or only very little amount, very less amount of nucellus is present. So such type of ovule is known as Tenuinucellate ovule. Okay, ovule in which nucellus is present in the large amount. Okay, the ovule in which this area is completely filled with nucellus is known as crassinucellate ovule. Okay, so this is called crassinucellate ovule. So there are two types of ovule based on the amount of nucellus present in them, Tenuinucellate and crassinucellate ovule. So this was actually the basic structure of ovule. Okay, remember the stalk of ovule is known as funicle, and the junction where stalk and the body of the ovule, they join with each other. That junction is known as hilum. One last point to note here is sometimes the stalk of the ovule, okay, 1 minute I'll change the color. Sometimes, for example, this is the stalk of the ovule. So sometimes the stalk of the ovule actually, little bit moves forward, or it actually little bit covers the part of the body. Okay, this is the stalk of the ovule. So it actually it will not stop only till the end of the stalk, but it will also cover some part of the body of the ovule. Okay, and this part is known as raphe. What it is called? Raphe. Now, if you have must have seen bean seed, the rajma seed. So this is actually the concave side of the rajma seed. Now, if you will observe this seed from the concave end, okay, if you will observe this seed from the concave end, so you can find out that at the bottom we have a we actually have a small opening which is called which is actually the opening of this micropyle, which is formed from the micropyle. Then we have very light color or white color scar, which is actually the scar of hilum. And then we have another very small scar. This scar is of raphe. Okay, so raphe is nothing but the extension of stalk on the body of ovule, the little extension of stalk on the top of the body of the ovule. Okay, so this can be easily observed in the bean seeds. Okay, the bean seeds, the rajma seeds which we eat at our home. Okay, so let's quickly revise the function of these parts of the ovule. The ovule is having following parts. The first part is known as attachment part. Okay, so attachment is having three parts. First one is funicle. So funicle is the stalk of the ovule. This is the stalk which actually attaches ovule to the placenta. Okay, so the stalk of the stalk of the ovule attaches ovule to the placenta. Okay, and the stalk is known as funicle. Second attachment point is hilum. So hilum is actually the junction between the stalk or the funicle of the ovule and the body of the ovule. Okay, so so hilum is actually the point from where this body of ovule arises after the funicle. Okay, or after the stalk of the ovule. And then we have raphe. Raphe is a ridge-like structure formed where funicle fuses with the body of the ovule. Okay, when funicle actually a little bit extends forward and covers the body of the ovule, and it forms the ridge-like structure which is called raphe. Next is integuments. There are two types of integuments present in the ovules, outer integument and inner integument. Okay, so remember these integuments, these are the group of cells and they form the protective covering around the mass of nucellus and embryo sac. Okay, this integuments, they actually originate from the chalazal end. This is important to note here. Integuments, they originate from the chalazal end and they lie opposite to the micropyle. Okay, they originate from the chalazal end, and the end which is completely opposite to the chalazal end is known as micropyle. Okay, so micropyle is actually the pore-like structure on one side of the ovule. Okay, one side of the ovule is having a pore-like structure which is called micropyle. Okay, and in micropyle the integuments are absent. Next is nucellus. Nucellus is a mass of cell that lies enclosed within the integuments of the ovule. This we have already seen in the previous slides. Nucellus is actually the mass of cell and it is present inside the integuments, okay, integuments of the cell. And the cells of nucellus, they have abundant reserve of food material. This is very important point. This is actually the function of nucellus. The cells of nucellus, they are like, highly rich in the amount of food present in them. Okay, they actually have abundant reserve of food material. Okay, and they actually provide nourishment to the embryo developing inside them. Okay, and this we will learn maybe after fertilization, in post fertilization events, that after the process of fertilization, the cells of nucellus, they degenerate. Okay, and they actually form covering around this developing embryo, which is actually known as perisperm. Okay, so this nucellus forms the perisperm. After after the process of fertilization, this the cells of nucellus, they degenerates. Okay, they degenerate and form perisperm. Okay, after the process of fertilization, so they form perisperm of the seed. And this I have already told you that there are so many cells in the nucellus. So one of the cell of the nucellus, it develops into the megaspore mother cell. This megaspore mother cell is diploid, and later on this megaspore mother cell undergoes the process of megasporogenesis and gives rise to the haploid megaspores. Okay, and the last part of this nucellus is embryo sac. Embryo sac can also be called female gametophyte. Okay, so this female gametophyte is actually covered by a thin membrane which is called embryo sac, and it is located in the nucellus. And then an ovule generally has a single embryo sac formed from megaspore through reduction, division, meiosis. Okay, this we are going to learn in detail in the next video. Okay, so embryo sac or female gametophyte is present inside the, it is actually present or enclosed inside the nucellus, and generally one ovule always has only one embryo sac. Okay, and this embryo sac is actually formed from the megaspore mother cell. Okay, from the megaspore mother cell through meiosis division. Next video we are going to learn about the process of megasporogenesis and the development of female gametophyte.